Okay, so let's start with a number that's, well, it's almost impossible to really wrap your head around. 2 meters. That is the length of your entire genetic instruction manual, your DNA, if you were to stretch it all out, end to end.
2 meters. It's staggering.
And now you have to imagine folding that 2-meter string
Yeah.
into a package so incredibly small. It fits inside a cell's nucleus, which is only about 10 micrometers wide.
Right. And that's what we're getting into today. It's not just about packing something away. It's about organization under immense pressure.
So this deep dive is all about that architecture, the chromosome.
Exactly. Our sources map out 3 really critical areas. First, just how that DNA gets compacted in the first place. Second, how the ends of it are protected. This gets into telomeres and even aging itself. And finally, what happens during cell division, when things go catastrophically wrong.
That scale is what gets me. A 2-meter string that's only 2 nanometers wide. It just highlights that the cell needs more than, a junk drawer to shove it all into.
That's a great way to put it. It needs this incredibly precise, multi-tiered system.
And it has to be accessible, right? Not hopelessly tangled.
Absolutely. The cell has to achieve this 1,000-fold reduction in volume, but still be able to rapidly unpack specific genes to read the instructions and then pack them right back up again.
Okay, let's unpack that process. Let's start at level 1. What's the very first step in this massive shrinkage?
So the initial step is all about stabilization. The DNA double helix wraps around a core structure made of 8 proteins called histones.
Histones.
Right, and these are alkaline proteins. They have a positive charge, which strongly attracts the negatively charged backbone of the DNA.
So it's a magnetic attraction, basically.
It is, and this forms a kind of bead-like complex that we call a nucleosome. This first step alone shortens the DNA by about 7 times.
Okay, but how does the cell make sure all 3 billion of these base pairs find those histone cores without just creating a huge mess? Is it passive or is there something managing it?
It's very highly regulated. The basic attraction is, electrostatic, but the positioning is critical. The cell uses these specialized remodeling complexes that can actually slide the nucleosomes along the DNA.
To expose certain genes.
Exactly, to make sure that crucial gene sequences are either exposed for use or covered up to be silenced. And this whole structure, the DNA wrapped around the histones, is what we broadly call chromatin.
So we've got these beads on a string, the nucleosomes. What's the next level of folding?
The nucleosomes and the linker DNA between them, they coil together. They form this tight spiral structure. We call it the 30-nanometer chromatin fiber.
Okay.
And that takes the whole structure to about 50 times shorter than its original length.
50 times.
But here's where we need to distinguish between 2 main states, which is critical for how genes work.
Right. This sounds important. What are they?
They're called euchromatin and heterochromatin. Euchromatin is the looser form. It's more accessible, where genes are actively being transcribed. Heterochromatin is the opposite. It's highly condensed, tightly packed. Genes there are usually silenced.
So even in a non-dividing cell, when the DNA is in this looser chromatin form, it's not just a messy bowl of spaghetti in there, is it? How does it keep all 46 chromosomes separate?
That's a fantastic question, and the answer is something called chromosome territories. Even when it's not condensed for division, each chromosome hangs out in its own particular non-overlapping area of the nucleus.
Like its own neighborhood.
Exactly. It limits entanglement and helps with gene regulation, and that organized state is what we mean when we say chromatin.
But when the cell is ready to divide, that's when it needs maximum compaction. It all has to be visible.
Precisely. Before division in what's called the S phase, the DNA replicates. So you get 2 identical copies. We call these sister chromatids, and they are bound together very tightly, especially at this central region called the centromere. And this final super compact structure is what you can actually see under a light microscope. It's the ultimate transport vehicle.
So we've mastered the architecture of packing the DNA. Let's shift to maintenance. How do you stop the ends of this long linear molecule from, well, fraying?
Right.
This is where telomeres come in, our protective caps.
Telomeres are absolutely vital, and they exist because of something called the end replication problem. It's just a consequence of how our DNA copying machinery works. The enzyme, DNA polymerase, it physically cannot copy the very, very end of the DNA strand.
So without some kind of protection, the chromosome would just get shorter with every single division.
It would. It would start chewing away at actual functional genes.
So the cell essentially builds in a disposable buffer zone.
That's a perfect analogy. Telomeres are just sections of highly repetitive DNA sequence. In humans, it's TTAG, over and over, that act as this buffer. And the shortening is very real. The average telomere length drops from about 10,000 base pairs at birth to maybe 4,000 by the time you're 100. It's a literal biological clock.
What really stands out in the sources is that this decay isn't just a set rate. It's heavily influenced by external factors. Why are telomeres so vulnerable?
Well, the sequence itself, being so rich in guanine, is just inherently fragile. It makes it really susceptible to damage from oxidative stress.
From the environment, our diet, that sort of thing.
All of it. And then there's chronic inflammation, which dramatically accelerates telomere loss. Certain inflammatory signals in the body directly drive this shortening and push the cell toward what we call senescence.
And when those protective caps get too short, what's the final outcome for that cell?
The cell has a very dramatic reaction. It sees those unprotected ends as a catastrophic DNA break.
Like an emergency signal.
A huge one. It triggers a powerful damage response, which leads to something called replicative senescence, basically permanent retirement. The cell cycle arrests, and eventually the cell undergoes apoptosis, or programmed cell death.
So how does some cells get around this? I'm thinking of stem cells, or more alarmingly, cancer cells. They seem to be immune to this clock.
They activate the ultimate counterbalance, an enzyme called telomerase.
Telomeres.
It's a special kind of enzyme that basically functions as a reverse transcriptase. It carries its own little RNA template, and it adds those TTG repeats back onto the ends of the chromosomes.
So it's rebuilding the buffer zone.
It's actively rebuilding it. This is why it's so active in stem cells, which need to divide a lot, and why it's pathologically active in about 90% of cancers. It gives them a form of immortality.
But telomerase isn't the only defense mechanism, is it? I think the sources mention a physical shield as well.
That's right. The second line of defense is the shelterin complex. This is a group of 6 proteins that physically bind to the telomere DNA. They form a protective cap. Sometimes it's described as a T-loop structure, and it literally cloaks the chromosome ends.
So it hides them from the cell's own surveillance systems.
Exactly. It prevents the cell's damage repair systems from mistakenly thinking the natural end of the chromosome is a break that needs to be fixed.
This is fascinating. The sources really position telomere length as one of the best biomarkers we have for biological aging. So if it's measurable, are we looking at ways to influence the speed of this clock?
Oh, absolutely. The research really falls into 2 camps. First, there are lifestyle modifications. Things like antioxidant-rich diets, exercise, stress management, they all help mitigate the oxidative stress and inflammation we talked about. And second, there's a huge interest in pharmacological agents. Some are activators, like a compound called TA65, that aim to boost telomerase activity. And on the flip side, there are inhibitors, like a drug called imetelstat, which is being studied to try and kill cancer cells by turning their telomerase against them.
An incredible biological maintenance project. Okay, let's shift focus. We talk about chronic decay, but what about catastrophic one-time errors? What happens when cell division itself goes wrong?
For that, we turn to a technique called karyotyping.
Right.
A karyotype is just a standardized, organized picture of an organism's chromosomes. They're arranged in homologous pairs, one from each parent, lined up from largest to smallest.
And scientists look for specific markers, right, like banding patterns where the centromere is.
Exactly. It's a very systematic process. A normal human karyotype shows 46 chromosomes, 22 pairs of autosomes, and that 23rd pair, the sex chromosomes. It's our main tool for spotting abnormalities.
And what's the fundamental mechanical error that causes most of these numerical disorders?
The mechanism is called nondisjunction.
Nondisjunction.
It simply means the failure of chromosomes to separate properly during cell division, specifically during meiosis when we're making sperm and egg cells.
And the sources are very clear that when this error happens, makes a huge difference to the outcome.
It makes all the difference. If nondisjunction happens during the first meiotic division, meiosis the first, well, that's the more damaging event.
Why is that?
Because the homologous chromosomes fail to separate. It results in 4 abnormal gametes. 2 will have an extra chromosome, and 2 will be missing 1.
So all 4 potential offspring from that division are affected?
Every single one. But if the error happens later in meiosis II, the outcome is split.
Okay.
In that case, the sister chromatids fail to separate. You end up with 2 normal gametes, 1 with an extra chromosome and 1 missing a chromosome.
And when one of these abnormal gametes is fertilized, you get either a trisomy, an extra chromosome, or a monosomy, a missing one.
Correct. A trisomy is 2n plus 1, and a monosomy is 2n1.
When we look at the specific syndromes, there's a real spectrum of severity. Why is a trisomy of chromosome 21 Down syndrome compatible with life, while others are almost always lethal.
It generally comes down to size. The larger the chromosome, the more genes it carries, and so the more catastrophic the imbalance is when you have an extra copy. Chromosome 21 is one of our smallest, so while Down syndrome presents challenges like intellectual disability and heart defects, the gene dosage imbalance is survivable.
Whereas something like trisomy 13.
Patau Syndrome, yes. That involves a much larger chromosome, and the massive gene imbalance leads to severe systemic failures. It's often lethal within the first few months of life. The body just can't tolerate it.
And it's important to note, not all of these disorders are about a whole extra chromosome. Some are structural.
That's a great point. A classic example is Patau Syndrome. It's not a trisomy, it's a deletion. Part of the short arm of chromosome 5 is just gone.
Which causes that distinctive cat-like cry in infants that gives it its name.
Exactly. Along with severe intellectual disability, it shows that even a partial loss can have profound effects.
Okay, let's move to the last piece of this puzzle, the 23rd pair, the sex chromosomes. This presents a really unique biological problem, dosage compensation. Females have 2 X chromosomes, males have 1 X and 1 Y. So how does the cell prevent females from making double the amount of all the proteins coded on the X chromosome?
This is solved by a really elegant mechanism called X-Inactivation. It's also known as lionization, after Mary Lyon, who discovered it. Early in development in female mammals, one of the 2 X chromosomes in every cell is randomly chosen to be silenced. It gets packaged up into that tight heterochromatin so it can't be read.
And once that choice is made in a cell, all of its descendants stick with that choice.
That's right. And it results in what we call mosaic expression. The classic example you always hear about is the calico cat.
Right, with the patches of orange and black fur.
The genes for those colors are on the X chromosome. So different patches of cells shut down different X chromosomes, and you get that beautiful mosaic pattern in their fur.
And you can actually see the evidence of this silenced chromosome in the nucleus, can't you?
You can. It condenses into a little dark spot called a Barr body.
A Barr.
And the whole process is controlled by a specific region on the X chromosome called the X-Inactivation center, which produces a special RNA molecule called ZIST. This RNA literally coats the X that's going to be inactivated and triggers its silencing.
Okay, so if this system is so effective at balancing the dose, why do we still see syndromes when people have atypical numbers of sex chromosomes, like Klinefelter, which is XXY, or Turner, which is just XO?
That is the million-dollar question, and the answer is what I'd call the escape clause.
The escape clause.
The inactivation isn't 100% complete. In humans, somewhere between 12 and 20% of the genes on that inactive X chromosome actually escape. They remain active.
I see. And a lot of these escaping genes are in regions that are also present on the Y chromosome.
Exactly. They're in what we call pseudo-autosomal regions. Since they're present in both sexes anyway, they don't really need to be silenced.
So if you have Klinefelter syndrome, XXY, you're getting 2 active doses of all those escaping genes instead of the usual one.
Precisely. And that dosage imbalance leads to the traits we see: tall stature, sterility. Conversely, in Turner syndrome, XO, you're missing an X, so you only have 1 dose of those crucial escaping genes, leading to short stature and other issues. It's the only non-fatal monosomy in humans, and it's because the imbalance is limited to that smaller subset of genes.
This entire journey has been just breathtaking. From the sheer engineering of packing 2 meters of DNA into a nucleus, to maintaining the ends with telomeres, to the high-stakes precision of cell division.
It really synthesizes the core requirements for life, doesn't it? You need stability, you need manageability, and you need precision.
Yeah.
The chromosome is the central piece of architecture that makes all of that possible.
For me, the most compelling part of this whole deep dive is the link between the chromosome and the process of aging. The telomere is this measurable cellular timekeeper. We know its decay is influenced by lifestyle, by stress, by inflammation. We even have researchers developing enzyme activators to influence it.
We do.
The fact that the very mechanism governing how long a cell can live and divide seems so accessible, so malleable, it suggests a really profound target for intervention. So the question to leave you with is this: if we can truly modulate the speed of that cellular clock, what are the ultimate implications for human health and for our entire relationship with age-related disease? That's all the time we have for this deep dive. Thank you for joining us.